Well, I'm really pleased to be here. Thank you very much for treating this topic as important as it is. My lab has worked on this for ages, and it's a very frustrating topic, but I'm very, very glad that you're focusing on it. So one of the things I want to talk about first is some definitions, because I think they get us into trouble. What do we mean when we want to prevent metastasis? And we have a semantic problem. And the problem is that metastasis is a noun that has two meanings. The first meaning is the spread of disease, such as cancer, from an initiating site to a distant site. So it's the process by which this spread occurs. The second meaning is the secondary malignant tumor that results from metastasis. So it's both a process and the product of that process. And that gets us into trouble when we talk about preventing metastasis. So what do we really need to do to prevent metastasis in patients? And I think that the important thing is we need to prevent some step or steps in the process with the goal of preventing the formation of the product of that process. So it's often said, well, why are you bothering studying this? The horse is already out of the barn by the time many breast tumors are diagnosed. This is true up to a point, but I think that it's leading us astray. It's really not a helpful way to look at things, because what we really want to do is prevent clinically relevant metastases. And I'm going to define that as a metastatic tumor that grows to cause irreversible physiologic damage to a vital organ. That's what matters. That's a lethal metastasis. So how can we prevent metastasis? First question, can we prevent metastasis, the noun, the process? And the answer to that is yes and no, because parts of the process have already likely occurred long before you know that you have breast cancer. Cells are known to leave primary tumors early in the course of disease, but we don't need to prevent all of the steps in the process. What we need to do is prevent clinically lethal metastases. So we need to prevent the growth of metastases from causing irreversible physiologic damage to vital organs. So up until the point when that has happened, we do have a therapeutic window. We just have to learn how to do it. And then all in quotes, we need to do is prevent the latter stages of the process, because that's what kills patients. So what are some of the steps in the process of metastasis that gives rise to these lethal metastases that might be targeted? Now, we've heard some very nice work about preventing tumor formation in the first place, and that is the place to go. I mean, if you can prevent a primary tumor from forming, then you prevent all of the later steps. That would be the fine goal. So we've heard a bit about prevention, or really risk reduction, of primary tumor formation, lifestyle factors that Dr. Willett talked about, which may reduce risk, but as you heard, they're complicated. And then various prophylactic ways of treating women at risk, high risk. So these are healthy women. The STAR trial, various hormone manipulations, prophylactic mastectomy for women at particularly high risk. Lifestyle factors are one thing, but these interventions obviously have risks that you have to balance against the benefits. But if you could prevent the primary tumor from forming, you would indeed prevent the later steps from happening. The problem with that, though, is that's not going to solve the whole problem, because it's unlikely that we're going to ever be able to prevent all breast cancers. There are women at particularly high risk. It would be nice if we could prevent them. And as Maria pointed out, some patients do present with metastases at diagnosis. So we've got to learn how to deal with this what's going on in the secondary organ. So where are there opportunities for interventions in the process in order to prevent the formation of lethal metastases? As I said, some of the earlier steps probably have already occurred by the time of diagnosis, but if they're ongoing because of reseeding, they may still be intervention sites. Work that my lab has focused on is tumor dormancy, which is a huge issue for breast cancer. And the two strategies there are, can we figure out how to maintain cells in a dormant state, then they wouldn't be an issue, or can we actually try to kill them? Right now we think that they're resistant to at least cytotoxic chemotherapy. Little micrometastatic disease is essentially what's the target of adjuvant therapy. and we know that that works. So we know that you can prevent the progression of metastases to form vascularized, clinically detectable lethal metastases. And we know that long-term hormonal therapy also helps in this setting. So there are certainly steps that have been shown to be successful in preventing the progression to lethal metastases. So how are we doing? I thought I'd bring some Canadian statistics since I work in Canada and you all have access to the US statistics. So this is from yearly Canadian statistics from 2011. And this shows the age-standardized mortality rates for breast cancer. And it's the curve that gradually goes down a little bit. It's going down, but it's gradually going down just a little bit. So if you look at the actual figures, we've had a 30% decrease in age-standardized mortality from breast cancer from 82 to 2011. This is clearly not good enough, but it means that the problem can be attacked. So it gives me hope that if you can get it going in the right direction, get it going faster, we can make more progress. So what do we really need to do? This is what guides my whole lab's effort. First of all, we know that most breast cancer deaths are due to metastasis. So if we're going to reduce breast cancer mortality and morbidity from treatment and from the disease, we have to better understand the process, and we really don't understand the process well. And it also means that new treatments need to be assessed for their ability to work in the setting where we need them to work, either to prevent, delay, or treat metastatic disease. And this is not how drugs are developed currently. So I thought I'd show you two little examples with not much detail of what my lab is doing, just as examples of how we're trying to approach the problem. First of all, we have an early breast cancer progression project that's trying to identify genes that regulate progression from early breast cancer, atypical ductal hyperplasia, ductal carcinoma in situ, to invasive mammary carcinoma, because that's the one that can then metastasize. We're doing this both to develop prognostic biomarkers and also for therapeutic targets. And the reason here is shown from the 2011 progress report. And the main problem is that with early breast cancer, not all of them will progress, but we're treating them aggressively because we can't tell the difference between those that will and those that won't. So the way we're approaching it, and again, no details, we have a set of 3 cell lines from one woman who had metastatic breast cancer. And when you put these cells either into mice or into lab tissue culture, they mimic 3 stages of early breast cancer progression. One of the lines looks for all the world like atypical ductal hyperplasia. Another one looks like DCIS. Another one looks like invasive mammary carcinoma, and it can metastasize in mice. So we've done expression profiling. We have all of the genes that are up and down regulated, and then we're trying to pick genes that we think are functionally important, and then we're changing their expression levels in the cells to try to see whether some of them are actually drivers of this progression, some of them which might predict whether a DCIS is going to be a bad player or a good player. So that's one approach that we're taking. The other area that we're spending a lot of time on, and which has been really frustrating, is trying to understand tumor dormancy. I mean, this is crucial for breast cancer. Dormancy can last for 20 years. That's a really scary thought. And we really understand very little about it. The extent of what was in the 2011 progress report is this one paragraph. And that reflects what we know about it. We know very little about tumor dormancy and then why or how late recurrences happen. So we've got to understand that. So in the context of a DoD-funded Center of Excellence to Pat Stieg, we've been doing some mouse modeling of MRI, monitoring brain metastasis in a mouse. And so what we're doing here, we're injecting iron-loaded cells to the mouse brain, and then we're monitoring the whole mouse's brain over time by MRI every couple of days. So here I've colored the dormant cells, which are iron-containing, they haven't divided, as red. And you can see that a bunch of them arrive because we've injected them to the mouse's brain. And then very gradually, some of those cells will convert to what we colored green metastases. But they're existing in the sea of dormant cells. And I want to know whether that's what's going on in breast cancer patients. So we're trying to find strategies to figure that out. So another issue that I want to talk about was touched on a bit by Dr. Ewell this morning, and that's tumor heterogeneity and progression. Yeah, I'll get there. I will. So we know that a tumor block is a snapshot in time. We know that tumors are heterogeneous. Not all the cells are identical. And we know that tumors change. This is one thing that they do. They progress in response to therapy or just because in response to the host responses. So cancer is a complex and dynamic target, and I think we have to wrap our heads better around this as the key issue in breast cancer. So I wanted to show this as a not perfect analogy. We know how to deal with flu. We accept that it's an evolving disease. We accept that every year we have to go out, figure out where the next dangerous flus are going to be. We develop a vaccine, we vaccinate people, and we repeat annually. Breast cancer is not, it's an evolution within a person, but we so far have been identifying a molecular defect, inventing or identifying a drug, treating as early as possible, and then repeating as necessary. In order to do that better, I think we really have to understand more about the biology of metastasis and the nature of this evolution. So I wanted to show a few things that are giving me hope that we're going in the right direction. This study was in kidney cancer, and it's a bioinformatics study that is reporting on the dynamic heterogeneity and evolution of tumors. It's going to be true in breast cancer as well. So what they did is they did a genomic analysis, mutation analysis on a kidney tumor, multiple regions of the kidney tumor, and various metastases. And what they found was that they were different and that they could make an evolutionary tree of how the tumor had evolved even within the primary tumor. Not all the sections were the same. And then the metastases, not all of them were the same. They have different molecular defects. If we had drugs that targeted those or understood how this heterogeneity developed, I think that would help. Two studies which both came from Canadian labs within 2 weeks of each other, so I'm pleased with that, give me hope that we're going to understand the complexity that's there. We're going to understand the evolution of what's going on. So the first study showed that for a bunch of triple-negative breast cancers, they were at different phases of their molecular evolution when they were identified. Some were more progressed and more complicated, some were more simple. The other study looked at 2,000 breast tumors and divided breast tumors into 10 new subgroups. They didn't track perfectly with luminal A's and B's and basals, that they encompassed all of them, and they had defined chromosomal defects. So somehow we have to understand those. So let me sum up with progress. I think we have made progress. We have shown that progress can be made. It's slow, it's frustrating, but we're making progress on a really complicated set of diseases. Earlier detection, I think, is more than just lead time. I think you're catching things sometimes at earlier stages of progression, which I think is good. We're getting better at preventing micrometastatic disease from progressing with things like adjuvant therapy. We have this ability to read the complicated bioinformatics of tumors, and we have to learn how to exploit that, but I think it's going to be a wealth of information. Challenges: why is metastatic disease so hard to treat? We know a defect that cancer changes. As we heard this morning from Dr. Willett, are there modifiable or lifestyle changes that could affect metastatic progression and growth? Or is whether a tumor will progress or not hardwired into the tumor? It's got to be a mix of the two, but we don't know where the mix lies. We're developing an evolving understanding of breast cancer subtypes. We don't have it nailed down perfectly yet, and they will change. And we need to know how to translate bioinformatics to actually benefit patients. So this is what I think we need to do. First of all, we need to figure out who is at risk for recurrence. How common are dormant cells? We don't know. Does everybody who's ever had breast cancer have dormant cells that mostly don't matter? Or is it only a subset of patients? What regulates whether those cells wake up or just stay dormant for the rest of your life? So that means we don't know who to treat, with what, and when. And then we have to balance the benefits and toxicities of long-term therapy, such as the long-term tamoxifen therapy, with the benefits. We know that biologically the target is there, but you have to balance risks and benefits. And then I think we have to understand why it is that metastatic disease is so darn hard to treat. And I think it has to do with a better understanding of the heterogeneity between tumors and within tumors and their metastases. We need improved therapies that target different subtypes, and the subtypes are evolving. We need to better appreciate how tumors evolve and how we can actually use this. This is what cancers do, and I think we have to figure out how to use this. Metastatic tumors are a dynamic target, so how do we actually target this plasticity? And then obviously we need the resources to do this. So I'm glad for your interest in this topic, and I'm working as hard as I can on it, and I need your help. Thanks.